Hi, my name is Catherine, and I'm an undergrad at the University of Rochester. And this summer, I had the opportunity to participate in the NIH SenNet CUSP internship program with Dr. Jose Lugo Martinez at CMU. And this summer, I worked on characterizing senescence markers in heart cells. So to start off with the definition of senescence, cellular senescence is often defined as a state of permanent cell arrest, and it can be induced by several types of stressors, including DNA damage and oxidative stress. And some common hallmarks of senescent cells include secretion of SAS factors, which include cytokines, chemokines, and growth factors, along with morphology changes to the cells, such as an enlarged nuclei. During aging, these cells accumulate and contribute to chronic disease, which is why they are important to study. Besides lab-based hallmarks, in computational work in single-cell RNA sequencing data, senescence is often identified by examining whether genes in a senescence gene set are highly expressed in a cell, and heterogeneity in senescent cell markers across different cell types and organs can lead to some discrepancy across gene sets. Some common gene lists include the Friedman cell age, gen age, SenMayo, and Go term analysis. So this summer, I worked with precision cut heart slice data courtesy of the Gukar lab at Pitt. Here, doxorubicin was used as the senescence inducers, and heart samples were taken from the left ventricles. Some variables that we looked at were sex, age, and treatment. Either the cells were treated with the doxorubicin or the control media. So our research objective was to determine potential upregulated senescence markers in the heart. Our computational workflow consisted of starting with the RNA sequencing data and running that data through the NF-core pipeline, which aligns our RNA sequences with genes to obtain counts per gene. And using those counts, we can further analyze our counts using PCA log fold change volcano plots, flow enrichment, and deconvolution to get cell type proportions. So to start off, we use UMAP to look at how our different samples were related in space. And from our UMAP plot, we saw that samples of the same treatment were generally clustered together in the same space. Next, we looked at the log fold change expressions of different cellular processes. On these heat maps, these samples are grouped as female old samples, female young samples, male old samples, and then male young samples. Cells that are red indicate the upregulation of that gene in the dox-treated cells versus the controls. Blue cells indicate those genes were downregulated in the dox-treated cells compared to the controls. And we wanted to look at these common cellular processes since they are involved in some senescence. And here across the heat maps, we saw general upregulation in the fatty acid oxidation and Krebs gene list, along with the glycolysis gene list. We also looked at the log fold change analysis of several senescence lists comparing the dox-treated samples with the control-treated samples. So we looked at the cell age Friedman Go set along with SenMayo and a union list of all these genes, which weren't shown, but they follow a similar pattern. So we saw a lot of variability in the upregulation and downregulation of the gene sets, but we did see consistent upregulation of the senescence genes in the male old samples. Since we didn't see consistent upregulation in our senescence gene list, we next looked at P16 and P21 expression levels, since P16 and P21 are common senescence markers and play a role in cell cycle arrest. And here with the heat maps, we also didn't see consistent upregulation in P21 or P16. I also did some more exploratory data analysis, here comparing the old samples and the young samples with a volcano plot. So here, the red dots indicate the significantly upregulated genes, while the blue dots indicate the significantly downregulated genes. And when comparing the old versus the young, considering both male and female together, we saw a higher number of downregulated genes compared to upregulated genes. When male and female were separated, we saw that males had higher number of significantly upregulated and downregulated genes. We also did the same comparison looking at the dox-treated cells versus the control. And through Go enrichment analysis, we looked at each set of upregulated and downregulated genes separately. And we saw that the upregulated genes with the cells that were treated with doxorubicin were more in the extracellular matrix organization and muscle processes, while the downregulated genes participated in nucleosome components and myeloid cell differentiation. Next, we did deconvolution using SCADEN, which deconvolutes bulk single-cell RNA sequences. And here the heart cell atlas was used as the single-cell RNA reference. And we decomposed the samples to get different cell type proportions. And we first looked at our data in another UMAP space and saw how our different samples were related to each other. Here, the same color represents samples of the same treatment, and we saw that they were generally along the same axes. Here we plotted a heat map of our deconvoluted cell types with our cell types on the x-axis and our samples on the y-axis, with alternating samples of control and dox. From our deconvolution, we saw a higher proportion of ventricular cardiomyocytes, shown with the deeper redder colors, compared to the atrial cardiomyocytes, which confirmed that samples were taken from the left ventricles. Another thing we noticed was a higher proportion of the myeloids in the control samples compared to the dox, which matches with our Go enrichment that showed negative regulation of myeloid differentiation in our dox-treated cells. We next did deconvolution using senescent cell reannotation, which takes our single-cell reference data, our gene list, and selects the top 5% of cells in that gene list and reannotates that cell type as the original cell type with senescence. So on the right, we see a heat map of our cell type proportions with our senescence reannotations. And the fibroblast senescence, mural cell senescence, and endothelial cell senescence had the highest proportions. And here's a comparison of our senescence reannotated deconvolution compared to our original cell type deconvolution. So to summarize, we didn't see consistent upregulation in our senescence genes across all samples, and there was no significant change in senescence signatures from our deconvolution. But interestingly, we did see that proportion of myeloid cells go down with doxorubicin, and myeloid cells are a type of immune cell originating in bone marrow, which would be interesting to study further. And limitations include that senescence may not have been fully induced during the experiment since samples were treated with acute doxorubicin instead of long-term treatment. In closing, I would like to acknowledge the NIH SenNet Consortium, Dr. Jose Lugo Martinez and Young Jae at Carnegie Mellon, and everyone in Dr. Gukhar's lab at the University of Pittsburgh for all their guidance and help this summer. I really enjoyed this research and learned a lot about senescence and computational skills that I hope to take with me into future projects.